Hello, my name is Aditeya Biswas, and today I will be talking about Emmanuelle Charpentier, one of the leading scientists in the field of gene editing. She's best known for her work in developing CRISPR-Cas9, a revolutionary tool that has changed the way we think about biology and medicine. Charpentier was born in 1968 in France. She studied biology, microbiology, and genetics at the Pierre and Marie Curie University in Paris, where she earned her PhD in microbiology. After that, she moved around internationally, working at various research institutes in the United States, Austria, Sweden, and Germany. What makes her background stand out is how independent and persistent she was. She wasn't at the most famous universities or labs, but she kept pursuing her interest in bacteria and molecular genetics, eventually leading to her discovery of the CRISPR-Cas9 system, where she won a Nobel Peace Prize alongside her partner Jennifer Doudna for that discovery. For her hobbies, Charpentier is very artistic, and she enjoys music and creativity. She has stated that she loves doing dance and playing the piano. This shows up in her approach in science. She often describes her experiments as a kind of creative process where she combines curiosity with innovation. And that's why she's been called an artist in gene editing for those interests and hobbies. What makes me admire Charpentier's story is her persistence. She's worked for years moving between different labs before her biggest discoveries, and as I stated, they weren't the most highest level labs or universities, but she still eventually discovered one of the most famous scientific inventions. This inspires me because it shows that success doesn't come overnight. You need patience and dedication, which Charpentier is a perfect example of. Charpentier's field of biology is microbiology and molecular biology. She focuses on how bacteria defend themselves against viruses. Normally, when viruses attack bacteria, the bacteria have to fight back or else they get destroyed. Her most important research looked at a system called CRISPR, which is in short for clustered, regularly interspaced short palindromic repeats. These are repeating DNA sequences in bacteria that store bits of viral DNA. It's basically like a memory bank of past infections, and when the same virus attacks again, the bacteria uses a system alongside a protein called Cas9 to recognize and cut the virus's DNA. By studying this system, Charpentier discovered a way to repurpose that system and use it for gene editing, and now scientists can cut any DNA sequence we choose in any organism. This leads to her highly cited foundational paper. In this scientific paper alongside Jennifer Doudna, she basically describes what CRISPR-Cas9 is and how it works as a programmable DNA cutting tool. And it is one of the most cited biological papers ever. And in this figure, what basically happens is, starting in the letter A, the bacteria produces two RNAs, a guide RNA and a tracer RNA that join together. Then these RNAs direct the Cas9 protein to the matching DNA sequence of a virus. And then the Cas9 cuts the DNA at that exact location, stopping the virus from working, which is exactly what happens in the letter E. What makes this revolutionary is that scientists can now design the guide RNA for any sequence they want and just cut DNA anywhere, and not just in bacteria, but in humans, plants, and animals, giving scientists a huge new tool that they can use to edit basically the code of life. For what basically Charpentier discovered or uncovered is, again, CRISPR-Cas9, which its applications are just used everywhere. Scientists can now knock out genes to see what they do. For example, in medicine, it's being used to fix genetic diseases at the DNA level, such as sickle cell disease or cystic fibrosis, or diseases of that sort. And in agriculture, CRISPR is used to make crops more resistant to diseases and pests. So with that, Emmanuelle Charpentier's persistence and creativity led to one of the most important breakthroughs in modern biology, and her discovery of CRISPR-Cas9 is already transforming science and its potential to change medicine and agriculture and just biology and the way we understand life itself. Thank you so much for watching. These are my references.